First time opening the Zoom meeting.
Okay, I'm going to,
لكم جوان الوقت.
بلديات زارين المستشفى وصيدلية معها.
بايلا بايلا.
لا حاجة واحدة منها. باي. بس في الزوم.
all right, I'm going to share my screen with you. Could you mute your mic? I'm getting,
بس سيارتي.
Linda, can you please mute your microphone?
Sorry.
Thank you. Can you please mute your microphone?
Microphone.
yeah, please mute it. You're getting background noise through your mic.
It's in the bottom left.
Do you all see the structure of life?
Yeah.
Yes.
Okay. That's what we're going to talk about for most of this period. Prokaryotic cells, eukaryotic cells, and viruses. What is a cell? Smallest unit of life. The cell theory, make sure you know this. Mark it down and have this well ingrained in your mind because it's going to come up again and it's very important that you know it. It has three parts. All living things are composed of one or more cells. The cell is the basic unit of life. There's nothing more fundamental that is living than a cell. And importantly, cells arise from pre-existing cells, in contrast to the idea that there can be spontaneous generation of life from non-living sources. So those three elements are components of the cell theory. And it is a theory because it is very well established. To my knowledge, there's never been an exception to any of these three elements. Also importantly, remember that viruses are not cells. They're sometimes described as acellular. They're not considered living because they cannot reproduce alone. They need a host cell in order to reproduce themselves. And we'll talk about that a bit more later. How do we see very small things? The scale of size. What you see here is a size scale from 1/10 of a nanometer on the far left to 100 meters on the far right. At the bottom, you can see that a meter, which is about 3 feet long. A meter is 100 centimeters. It's 1,000 millimeters, 1,000,000 micrometers, and 1,000,000,000 nanometers. So starting at the level of a nanometer, small molecules can be as little as a billionth of a meter. Larger molecules like lipids and proteins can be up to 10 nanometers, 10 to 100 times that size. When we move up the scale to viruses, they're about 100 nanometers, whereas organelles that we find within cells, mitochondria and chloroplasts, they're about a micrometer in size, roughly in that range. Most bacteria are slightly larger than that, although organelles and bacteria are in the same range of size from 1 to 10 micrometers. And then we get to the cells, the eukaryotic cells. Animal cells and plant cells, they're about 10 times larger than bacteria. The largest single cells, frog eggs are among the large ones, but ostrich eggs are even larger than that. And I've put a little picture here of an ostrich egg compared to a hen's egg. It's much, much larger, about 6 inches long, and weighs a kilogram or so. So the ostrich egg is just an example of how large a single cell can be. A single cell can be microscopic or it can be huge. But I should point out that ostrich eggs are not the largest cells known. For example, neurons in the neck of a giraffe are very large. One cell can be as long as a giraffe's neck, which is several feet. There are also algae that have cells larger than ostrich eggs, but just for a visual reminder of how big a cell can be, think of an ostrich egg. All right, then you have birds and humans, which are 2 to 3 meters, and trees, which can be as high as 100 meters. So this is the world that we perceive. Some of it, from a millimeter to 100 meters, we can see with an unaided eye, with no microscope. But to see things smaller than that, we need microscopes. And microscopes fall into several categories. There are light microscopes and electron microscopes, and these are used to see different things. Start with a compound light microscope. You see a picture of one here on the lower left. And on the right, you see 4 cells. These happen to be plant cells. And so if you can visualize them with a light microscope, because the light from the microscope passes through the cell and hits your eye. You can see some objects. Anyone know what you can see with a light microscope in a plant cell? Anyone want to guess? what those things are?
My name's Katrina Vibard, and can't you see the nucleus in a plant cell?
That large thing there is in fact a nucleus. And what are the smaller green things?
Patrick Dowell,
Uh-huh.
would those be chloroplasts?
Yes, those are chloroplasts. Plant cells are unique among living things in that they have chlorophyll. The chlorophyll is in chloroplasts. It's a source of all food on the planet. And we will talk a lot more about chloroplasts and photosynthesis later. But the point here is that with a light microscope, you can easily see plant cells. Electron microscopes. One type is called a transmission electron microscope. In this case, an electron beam is shown on the specimen. It passes through it, and then the image is picked up on a photographic film. And on the right here, you see the same 4 cells that you saw in the previous slide, but through a transmission electron microscope. And the point to be made is that transmission electron microscopy shows fine structure within cells. So what you can see here, this large object in each of the cells is the nucleus, and then you can see a lot more structures that a cytologist could name for you. We won't be concerned about that now. Just remember that TEM shows you the fine structure inside a cell.
Yeah, it looks more like an MRI picture than the first one, which showed the whole,
Looks more like an MRI.
to me at least.
Well, I don't know, maybe it does resemble that. The fact of the matter, it's a plant cell. And it's not an MRI, it's an electron micrograph. Okay, let's go on. Now, in contrast to a transmission electron micrograph, here's one from a scanning scope. And the difference is that with a scanning electron microscope, the electron beam hits the target and then bounces off, does not go through it. And so what that results in is that you get a very good look at the surface of a cell. You can see nothing inside it. And that's what you see here, the same 4 cells, but you see nothing inside them, you just see the surface, but you get a very close view of what is on the surface of these cells. So is that clear, the distinction between scanning and transmission? They're sort of the opposite. Transmission shows you the inside of a cell, scanning shows you the outside. And just for fun, I threw this scanning electron micrograph in, cat's tongue. Yeah, probably all of you have touched a cat's tongue. You know, it feels rough. It's almost like a file. And the reason is, you can see here, all the cells on the surface of a cat's tongue have a little tip on them. Now, we've been talking about microscopes. So they help us see little things, smaller than the human eye can visualize. Telescopes, in contrast, help us see very large things that may not be on this planet. So many of you probably have backyard telescopes where you can see other planets, maybe even some stars. There are very powerful telescopes, like the one on Palomar Mountain here in San Diego County. This is a very old telescope. It was started in about 1948. Now, for looking out into space, there are much larger telescopes, like this radio telescope on Arecibo Mountain in Puerto Rico. It can look far out into space, but the major constraint it has is that to get a picture of what's out there, it has to go through the Earth's atmosphere. And that's a big interference. So to get past that, there are now space telescopes, which are shot out into space. They can go on for a long time and travel great distances and take pictures of things without interference from the Earth's atmosphere. And in fact, they're used to study galaxies, and in particular, study planets within galaxies. Speaking of which, on the lower left, you see our galaxy, the Milky Way. There are many, many, many little dots there. Each of them is a star or a sun. And each of them has one or more planets. Now, the Milky Way galaxy is huge. At the bottom here, I give you some idea of how huge. It's, I should say that, I don't know if you can read it, but up at the top here, it says our sun. Our sun is located out toward the edge of the Milky Way galaxy. So one of those little dots is our sun. It's 200 light years in diameter. Well, what is a light year? Light travels at 186,000 miles a second. So a light year is how far light can travel in a year, not a second, in a year. So if you multiply 186,000 times the number of seconds in a year, you'll get the distance of a light year, which turns out to be 5.8 trillion miles. Okay, that's 1 light year. Now, the diameter of the Milky Way is 200,000 of those. 200,000 times 5.8 trillion. That's how many miles it is across this galaxy, which is not a particularly big one. There are much larger galaxies elsewhere in the universe. So in the Milky Way, there are about 300 billion stars. So if each one of them has at least one planet, that means 300 billion planets. Now, if looking from a space telescope, you take one image from one little section of the sky, you see what's shown here in the upper right. That's an image of one section of sky taken from a space telescope. And each point of light, regardless of size, each point of light is a galaxy. So within this one frame, there are roughly 15,000 galaxies. And if you imagine each of those is as large and as full of matter as the Milky Way, then you can, well, you can't imagine. It's just beyond our comprehension how much space and how much matter there is in the universe. I won't talk about it in this class, but I hope we have time at some point to talk about life. Is there life out there, various places in the universe? What kind of conditions have to exist in order for us to expect a life to occur? And also importantly, if there is life out there, what is the likelihood that we'll ever find it or that it might ever find us? Well, those are all interesting questions, and we have some data that help us think about them. And as I said, I hope we get to talk about those later. Any question about any of this before I go on? Because I'm going to leave space and come back to Earth very quickly.
I have a question, Professor
Yeah,
Patrick Dowell.
yeah.
I think it was the first slide. It was talking about cells. One of the 3 points, cells do not come from sporadically. So where did the first cell come from if they come from pre-existing cells?
That's a good question. There had to be a progenitor cell, right? There had to be a founding event. And there are a number of hypotheses about that. One of which is that came from another planet, came in on a meteor onto this planet. A more highly favored hypothesis is that life formed in the primordial oceans on this planet. As organic molecules began to form, and we know they did, eventually the chemicals involved learned how to make a membrane, and a membrane is essential for the formation of a cell. So after membranes formed and organic matter, organic molecules formed within them, there is a lot of evidence that life can form in a test tube in that way. And again, if I get a chance to show my slide set on life in the universe, I'll give you some numbers on what the likelihood of that might be. And the bottom line is, it's very likely that it happened on this planet. And if it happened on this planet, it probably happened on many others if the conditions are right. And so what these space telescopes show us is which galaxies have planets, have stars that have planets that are the right distance from the star, have likelihood of water and other things that we think are essential for life. We assume that life in the universe must be based on water. It could be based on something else, but no one has been able to imagine how. so far?
Professor, would you be able to go back to the very first slide? I missed that last bullet point.
Okay.
I know.
You're Rosemary Krolik, right? Okay.
Yeah. Thank
So
you.
you wanted to go back to which?
Just the first slide. I'm sorry. I know you post them, but I
The
just like
second
to.
slide, this one?
Yes, please. Thank you.
Yeah, it doesn't hurt to go over this again because it's very important that you know it. The cell is the basic unit of life. Nothing smaller than a cell is living. Viruses are smaller than cells, but they're not alive. Okay, and I can remind you that this slide set is in Canvas. You can access it. And this session is being recorded so you can go through it in real time as you study for tests and quizzes.
Professor, I have a quick question.
Uh-huh. Your last name?
Yun.
Okay, got you.
So for this PowerPoint, is that for the chapter 1 or chapter 2?
Oh, in the text?
Yes.
Does anybody have that? I don't have the numbers. But I think this one is pages 5 to 26 in the text.
Um,
Can anybody correct me if I'm wrong?
I think it's chapter 2, chemistry of life. If
No,
I'm not
this isn't
wrong.
chemistry of life. We'll go through a section on chemistry.
so it is chapter 2, right?
It's
No.
I believe
It
it
starts-
starts on page 27.
Starting on 27? Okay.
I believe so.
You'll find in the announcements all the page numbers that refer to the text.
Okay, thank you.
All right, we're back to little things. Cells can be divided into two general groups, either prokaryotes or eukaryotes. Prokaryotes are bacteria and archaea, whereas most eukaryotic cells are either plant or animal. And viruses are not cells. Focusing in on prokaryotes, they're very simple, usually just one cell, very small, 1 to 5 micrometers. The genome of prokaryotes is always DNA. So remember that. Yet, although they have DNA, they do not have a nucleus. DNA is found in what's called a nucleoid region, but it's not separated from the rest of the cell components by a membrane. And in fact, prokaryotic cells have no membrane-bound organelles. They do have organelles, for example, ribosomes, but they're not bound in a membrane. Bacterial shapes. If you find a bacterium, it will be some variation on one of these 3 basic shapes. Either a rod, shown in pink. These are known as bacilli, rod-shaped bacteria. They might be spherical, known as cocci, shown in the middle. Or they might be spirals, like spirillum, shown on the right. So let's consider the structure, the gross structure of a prokaryotic cell. If you look down on the lower left, bacteria and archaea have cell walls. They have ribosomes, which they use to make proteins. They have plasma membranes. All cells have plasma membranes. Getting back to your question, the first progenitor cell had to have a membrane to separate it from the environment. And prokaryotes have DNA, which is sort of diffusely spread out in the cytoplasm. And to give you an idea about this, a bacterial cell, E. coli in this case, is about 0.003 millimeters long. If you take all the DNA in an E. coli cell and stretch it out, it's about 1 millimeter long, about 300 times longer than the cell. So to accommodate that, the DNA is wound up, coiled in a little tiny knot so that it can be housed. And if it ever needs to be expressed, it has to unwind. The region of it that needs to be expressed has to unwind in order for it to be active. On the lower right is a scanning electron micrograph. Excuse me, not scanning, it's a transmission electron micrograph of a bacterial cell. So you can see that there's a lot of internal structure. The big light regions are the nucleoid, where the DNA is. The little tiny dots are ribosomes. Now we switch to eukaryotic cells. Eukaryotic, U means true, karyon means nut, which refers to the nucleus. Two general types, animal and plant. And again, the genome of eukaryotic cells is always DNA. The size is about 10-fold larger than prokaryotes. And it's believed that eukaryotic cells have evolved from prokaryotic cells. You can see that animal cells and plant cells, they resemble each other. The big objects in both cases are the nuclei, but notice that plant cells tend to have a regular box-like shape, whereas animal cells are very irregular. And you see that animal cells more strikingly shown here. Muscle cells on the left are long and thin. Here you see skin cells. Neurons are very thin. They often have a body from which projections extend out. These projections can be very long. As I said earlier, for example, in the neck of a giraffe, a neuron can be as long as the giraffe's neck, and it's a single cell. And of course, there are blood cells, which are very numerous in our bodies, white cells and red cells, which carry hemoglobin. And we'll talk about this later. That's how they transport oxygen. So here's a question. Let's think about this for a moment. In a human, there are about 230 different types of cell. And yet we have about 25,000 genes. Each of these 230 different types of cell has exactly the same genes. They all have the same 25,000 genes. So how can we explain how different cells with the same genomes can look and function so differently?
They're written differently, the genetic code.
Who's speaking?
Rosemary.
Rosemary. Okay. So what was your explanation? Rosemary?
I'm sorry, you cut up there for a second. I couldn't hear you.
Okay, so what was your explanation for this?
The genetic code's written differently, so it provides different kind of functions for the cell.
Okay, there's a thought. Anyone else have a thought?
Hi, my name is Abdurrahman Hamawandi. Just to get this right, you're saying that there's 230 different type of cells. They all have the 25,000 genes.
Yes.
I think that they have the same genes, so they can, when they function differently, they can like get together, like so they can function together. That's why they have the 25,000 genes.
Well, certainly the 25,000 genes provide all the functions that all the different types of cells need. But we still have to explain how, since all cells have the same genes, how can they have different functions?
I'm Justin Hansen. Maybe some cells use portions of the 25,000 genes. Each one uses like different portions of the 25,000, but they all have the same.
You're on the right track.
Maybe they grow differently.
They grow differently.
Yeah, maybe.
Patrick Dowell here.
Stephanie Arriola, sorry. Forgot to say my name before.
Okay, yeah, I got you.
Thank you.
Patrick Dowell here.
Uh-huh.
Similar to what Rosemary had said, I think possibly it could have to do with the sequence, that the order that they are in could determine what's going to be going on, maybe.
The sequence of genes on the chromosome, you mean?
Correct.
Possibly could be relevant to that. Any other thoughts?
Michael Soror here. You hear me? Hello?
Yeah?
I'm thinking maybe the shape of each of the cells, they don't have to be the same shape, so maybe their shapes, even though they're made out of the same material, could help them in different areas, like give them an advantage. Like,
well, it's true that cell shape can be involved in cell function, but we still have to explain how different cells deal differently with the same gene sets, with the same set of 25,000 genes.
Maybe the gene, they all have the same genes, but they're made up of different molecules, so that they form differently, or they work differently. I don't know.
Well, the cells certainly work differently. The genes are all the same.
My name is Zara and Nazeri. So basically, I think every cells has a different parts in themselves, and some of these cells have missing parts than the other ones. You know what I mean?
I don't know what you mean by missing parts.
So some of the cells maybe they have different parts than the others.
Well, they certainly have different cells to have different parts, but they all have the same genes. So we're trying to get at how that can be.
Taylor, real quick, there's 230 different cells with the same amount of genes. Could it be that they reproduce in different speeds? Some cells regenerate faster than others.
Okay, and the speed of growth or regeneration would be controlled by their genes. So fast regenerating cells would have the same genes as slow ones.
You can go first.
Oh, I'm sorry. Rosemary Crowley. I'm just gonna guess that the genes itself tell, like the genetic code tells how the cells, like how the cells are supposed to function, or like what makes up the cells. So there are different types of cells and different functions, if that makes sense. Sorry.
Well, what you said is true. There are different types of cells, and each different type has a different function. And what we're trying to explain is how you can get all these different functions with the same set of genes.
Hi, Kari Lopez.
Okay.
Maybe since they have the same genetic, whatever, some, like maybe some parts are stronger in cells and others are less stronger, so that helps them indicate what they're supposed to do.
Some genes are stronger than others. And what do you mean by strong?
Like they have a bigger impact than a cell that has to do like a specific function, and like for example another cell that doesn't do the same function, obviously maybe that genetic code is less overpowering over the other ones.
Someone had mentioned this earlier, but even though there's 25,000 genes, could the cell be using just partially some part of the 25,000 to do that function?
Yeah,
make it a stronger cell?
you're on the right track, and several of you have been coming close. In two words, the explanation is gene regulation. So you have, every cell has the same 25,000 genes, but no cell has them all turned on at once. Some cells have none of them turned on, and some cells have just a few. So if you think about development, say the fetus, if it's, say the fetus is making fingers, okay, it needs to turn on a set of genes, fingers. Once those fingers are formed, those genes turn off and they're never turned on again throughout the life of the organism. Once fingers are made, those genes are done. So this is how we get so many different functions, cell functions, from the same set of genes. That only a subset of genes is actually active at any given point in development. And so for example, if you're an adult and you injure yourself, you cut yourself with a knife, well, a bunch of cells have to start dividing, and they do that because the wound has sent a signal to a set of genes that says, okay, heal this wound. Okay, is that clear to everybody, that the key to understanding cell types is gene regulation?
Professor,
Yeah.
cells turn off or genes?
Say that again?
Cells turn off or the gene?
The genes turn on or off in response to signals from elsewhere. But if a gene's product is needed for some function, then that gene will turn on, make its product for as long as it's needed, and then it'll turn off.
Okay.
Don't worry a lot about this now. Just grasp the concept. We'll talk extensively about gene regulation and how it happens later on in the course. There are specific mechanisms that turn a gene on or off. And that if it's on, determine how fast it runs. Another question, why aren't we made of just one big cell? We know that single-celled organisms, they're always very small, but they get along just fine as single cells. We are very large organisms and we have many cells. So why is that?
Is it to reproduce? This is Bennett Taylor.
Uh-huh.
The cells keep knocking into each other and making other cells.
They do reproduce, right? They divide and keep making more and more, but why?
Like you were saying earlier, kind of to heal a cut. There's all kinds of different functions for multi-cell.
Yeah, there are different functions. The question here is why do you have to have so many cells in a large organism like us when small organisms can get along with just one cell?
Hi, in Didmar. It's because larger organisms tend to have organ systems and specialized functions of the body that requires more total specialized cells to perform those complex functions than a single cell with its organelles.
Yeah, you're getting at an important aspect of this. That's one of it. You need many different cells in order to have cell specialization. There's another important reason.
Yeah. Hi. So Katrina Vibbard. It's for volume perspective, where because we are a larger, like we'll have a larger volume with the same amount of like surface area as, it's kind of like the picture showing is where it's like we need multiple cells to have a larger volume ratio, even though we contain the same amount of surface area.
You're right, except it's the reverse.
Yeah.
Volume ratio. And so if you have, and you're right, the picture here shows the difference. With one large cell, there's a surface area of 5,400 square micrometers. If you divide that same volume into 27 smaller cubes, you now have 16,200 square micrometers. So you get a lot more surface area with the same volume if you have a lot more cells. And why is that important?
I have a dumb question, Professor.
No,
is it anything to do with like keeping, because we're made of a lot of water and it's keeping the water, I don't know, never mind.
Oh, that's not a dumb question, because what's one of the main things that cells are made of? Water. And water has to get transported. And it gets transported from cell to cell through cell contacts.
Okay.
And when later in the course, we'll talk about membranes. Membranes are so critical for life, and membranes keep cells apart, but yet they touch, and communication occurs at the points where they touch. So in the lower left here, I've got the two features: cell specialization, which you've mentioned, and transport and communication. Those are the reasons that we're not just one big cell. The animal cell structure, and I'm going to contrast in the next two slides similarities and differences between animal cells and plant cells. So if we start here on the left with the nucleus, it's purple, and the nucleolus is blue. Just outside the nucleus in all cells is what's called rough endoplasmic reticulum. It's a system of membranes where the ribosomes are. And the ribosomes are critical for making proteins. There are peroxisomes, there are mitochondria, various types of microtubules. All cells have a plasma membrane. The Golgi apparatus processes proteins. Lysosome is kind of like a garbage can. And animal cells have a centriole, and some of them have flagella, little tails that wag. Now, all of these things are found also in plant cells, except these three: flagellum, centriole, and lysosome. And if you look on the right, here's a transmission electron micrograph, which shows a good many of the features, the subcellular particles that an animal cell has. And they're all labeled. Now, if we look at a plant cell, sorry about this white blob here, but you can still see it. Many of the same features, except if you look on the upper right here, what plant cells have that animal cells do not have are a cell wall, a central vacuole, and a chloroplast. So why do plant cells need those and animal cells don't? Take a cell wall. Why do plant cells need a wall?
Patrick Dow here.
Okay.
Because they're typically rigid, kind of like they stand up straight, trees, stuff like that. They are, I guess, rigid.
Yeah, the rigid is a good word. Cell walls are made of cellulose, and it's a long polymer made out of sugar monomers, but it's very strong. And you're exactly right. That's why trees can grow tall, because they have walls and can support themselves. Why do plant cells have chloroplasts?
My name is Kaylin Hilberg, and I think that they have a chloroplast to help with photosynthesis, because doesn't the green?
Yeah, you're right.
Okay.
Chloroplasts have chlorophyll. Chlorophyll is the pigment that absorbs light from the sun, and that's the source of all food on this planet, chloroplasts. Okay, let's talk about the vacuole. Why? Animal cells have little tiny vacuoles, but plant cells have a huge one. If you look at the electron, or the electron micrograph here on the right, you can see that the vacuole occupies maybe half the volume of the cell. Why do plants need such a big one?
Because they store water on the plant. They store water on it.
They store
then-
water?
Yeah.
Yes, you're right. Vacuoles, they store a lot of things, but the main component of a vacuole is water. So why is that important to a plant? It's not important to an animal.
What was the question?
Why is a large volume of water within the cell important to a plant? It's not important to an animal.
Hi,
Hi,
Carlos.
go ahead.
It's because, well, an animal can look for water when needed and a plant can't.
Yeah, that's right. Animal, if it gets thirsty, it goes to the water hole and gets a drink. If a plant is thirsty, it can't move. It has to sit there and wait for rain. So like a camel, it stores water and its vacuole to hedge against a dry spell. Okay, good. We'll move on from there.
Hi, Professor.
Yeah.
Sierra Wishdale, I was just wondering what we were just talking about. Does that have to do with like natural selection, like how you know, animals can get their own water and plants can't, like just evolving?
Yeah, so you're right. A A vacuole has resulted from natural selection. So plants that, primitive plants that had vacuoles and could sustain themselves during droughts were fitter and were able to survive, whereas plants that didn't have large vacuoles became extinct.
Thank you.
Okay, now let's talk a bit about viruses. They're not cells. They're very important in biology because all forms of life have them. All types of cells have their virus parasites. Examples of viruses: tobacco mosaic virus, shown here on the left, T4 bacteriophage, and HIV virus as a lipid envelope. And you see scanning EM of each type here on the right. TMV is a rod. It's a sort of a stiff rod with protein on the outside and nucleic acid on the inside. T4 looks like a spaceship, attaches to cells with its tentacles. And HIV is a sphere. All viruses are composed of a protein coat that surrounds the genetic material. And now here's a difference. In contrast to the other types of cells we've talked about, viruses can have either DNA or RNA, depending on the type of virus. So if you consider these three, TMV has RNA, T4 has DNA, and HIV has RNA as its genetic material. And here you see coronavirus, which is very much among us these days. We're concerned about it. You can see a scanning EM virus particles, same thing in color. And at the bottom here, the virus attacking the virus, the little yellow dots, attacking a human cell. And on the lower right, you see drawings of both the surface and the interior of a coronavirus particle. And what you see, I've circled two things in red, the spike protein and RNA. The spike protein is a protein on the surface that is essential for the virus to infect a human cell. This spike protein attaches to the human cell. There's a specific receptor on the human cell it attaches to, and that's how it starts an infection. The other important thing is that its genome is RNA, and so there are genes in this RNA that encode for all the other components, including the spike protein. Now, you've probably all heard in the news about the vaccines that are becoming available. Two of them, from Pfizer and from Moderna, are RNA. They are nothing but a bit of RNA from the virus, not the whole thing, just the bit that codes for spike protein. That RNA is packaged in a lipid bubble, tiny lipid bubble, and that's the vaccine. It's just simply RNA in a lipid nanoparticle. And that's injected into your muscle. The nanoparticles go into the cells, the RNA comes out and is immediately translated from RNA into protein. And that protein then is the immunogen that prompts your immune system to be active then against the virus in the future. So when you hear about vaccines, the two most prominent ones right now are based on RNA. Not all of them are. AstraZeneca has just come out with one that has a completely different basis. We can talk about later. We're running out of time, so let's go through a couple of questions. You're looking at a cell under a compound light microscope. And this cell is about 50 micrometers in diameter. You can see its nucleus. You can see it has a cell wall. What type of cell is this?
And we plant cell
Why do you say that?
as a cell wall.
Cell walls, what are prokaryotes? So what else do you know about it?
It's the plant cell because it has the cell wall and it also has a nucleus. And eukaryote cells are the only cells with nucleus, or a nucleus. And the fact that it has a cell wall means it would be a plant cell.
Yeah, it has a cell wall and it has a nucleus and it's about the right size, 50 micrometers. So yeah, this is a plant cell. Let's move on. For another example, looking at a cell that's rod-shaped, about 4 micrometers in length, so it's smaller, does not appear to have any internal structures, but it does have a cell wall. So what type of microscope and what type of cell? It has a cell wall. You can't see anything inside it.
Is that like a scanning something electron microscope? I don't know how to say it. Something scanning.
Okay, it's 4 micrometers, which is pretty small. So if you were looking at it with a transmission electron microscope, would you see any internal structures?
No.
Remember what transmission does? It's very good.
Transmission, yes, transmission, you'll see the inside. Scanning, you'll see the outside.
Right. And so this doesn't appear to be either one of those. So it's probably a compound light microscope. So it's not showing the inside of the cell clearly, but it is able to show the cell wall.
It's a prokaryote.
So it's a prokaryote. That narrows it down. Okay, good. And finally, you're looking at a structure that's really tiny, 0.02 micrometers, doesn't have any internal structure, has no organelles. What type of microscope are you using and what is it?
Electronic microscope?
What type?
Oh, it doesn't have.
It's showing.
A light electronic microscope?
Yeah, but what type of electron microscope? It has no internal structures that you can see. So it's showing the outside of. And it's very small. So something that size would be what? This is on the size of molecular, the size range.
So a virus?
Yeah, it's probably a virus because it's so little. You're probably using a scanning because you can't see any internal structures. All you're seeing is the outside of it. And so it's probably a virus and it's probably a scanning scope. Okay, I think, yes, I don't want to talk about pancreatic cells at this point, and unfortunately we are out of time. So I'll stop at this point And we'll try to work in some of the things I didn't have time for in the future. And remember that on Wednesday, in addition to class, you have a quiz and you can take it anytime during the day and you will be allowed 15 minutes. And it covers the first 2 sections of the course, scientific inquiry and introduction to biology. Questions will be of the same type you saw on the practice quiz.
Professor, it's gonna be a multiple choice, right?
There are a couple of multiple choice questions. There are some true and false. There's 1 or 2 essays. There are 10 questions in all. They're all the sort of thing that you look at them and you either know the answer immediately or you don't.
Okay.
Professor, will you be posting, I'm sorry if I missed this, will you be posting the PowerPoint right after class today?
Yeah, it's there right now. This
okay.
is there now. And as soon as the recording of this session is processed, I'll send you an announcement with the website for it.
Okay, thank you.
Anything else before we go?
Question for the, you said, what does the quiz cover? One more time, sorry.
The first 2 classes, what we covered last week, which are methods of scientific inquiry and the introduction to biology. So if you study the slide sets, the recordings, and the text, references in the text, you'll be well prepared.
Thank you.
Anything else?
You had said at the end of the class, you were gonna get me the 2 papers for last.
Yeah, the 2 worksheets, and I'll give you access to the practice quiz.
All right, you can just email me the worksheets if you want.
Okay, I may do that. It might be the easiest thing for me to do is just send you an email with attachments. Although I can make the quiz directly available to you. I'll just.
Yeah, it'll be in Canvas.
So if you go into Canvas and click on quizzes, click on quiz 1, which is listed as a practice quiz.
All right. So I'll just wait for your email attachments and then I'll read up on it and then I'll take the practice quiz.
Yeah.
All right, thank you.
Sure.
Thank you very much, Professor. Have a great night.
All right. I will proceed to end this unless there's something else anyone has. Hearing nothing, I will end that and then end the meeting for all, and I'll see you all on Wednesday.
Good night, Professor.
Good night.
Bye.
Bye.
Bye.